Thank you very much. Okay, so obviously nothing that I do would be impossible, would be possible without many of these much more famous, brilliant other people that I work with, and I would like to put all of their names, and I tried, but this is how it looks. So really not much that I can do, and I'll show a couple of names at the end. So we are talking about computational processes in biology, and basically different processes that occur in the cell. So you have different kinds of models, and you have computational complexity and algorithmic issues, and it's a fascinating area between biology, control theory, discrete math, and computer science. So the modeling problem is that how do we incorporate the biological data? And so, for example, interaction data can be produced in the form of a diagram that people have seen mostly here, like Legel diagram, and we may want to pose questions about the network, about the dynamics of such a network, like how the behavior of a node depends on other nodes and so forth. And dynamics can be continuous and discrete depending on the state variables. Time variables can be continuous, discrete. It could be a hybrid model. It could be deterministic, probabilistic nature of the model. So, for example, on a very broad level, there are lots of basic research questions that are of interest, like, for example, which models are experimentally falsifiable, so that they can be discarded, how models differ in terms of the computational power, and how feedback loops affect the capabilities of different models, which is a very, very interesting topic, and what degree of parallelism is possible in different models. And if you are a computer science student, not so much into biology, but a little bit of biology, and if you work in distributed systems, these are some of the things that might interest you. Think of the cell system as a kind of distributed system. It has some nice, interesting properties, like for many cellular networks, there is no global clock, but then somehow the processes tend to synchronize. How? In distributed computing systems, you also have this kind of problem sometimes to handle this. And spatial effect, like localization. So, for example, for the World Wide Web, we have these geographical locations affecting communication and so forth. And so what is the corresponding rule for localization in cell behavior? What about the communication delays? How do they affect dynamics? And so forth and so forth. So in this talk, I'll provide a brief overview of research questions that my groups and my collaborators have looked at, mostly high-level overview, and technical details maybe skipped because of lack of time. So the one thing that I first wanted to talk about is reverse engineering. So basically, it's something that people in engineering, electrical engineering, and other places always do. You are given some knowledge about the process. You have access to some kind of experiments, in the case of biology, biological experiments, and you want to gain more knowledge about the system. So there are very many ways of doing it, and you can even find monographs or survey papers or books on that kind of stuff. So the only thing that, the one method that I'm going to mention is this modular response analysis method. And we are just going to describe the method, and followed by what is the computational question there and some kind of biological validation. So what you have is a differential equation model. So you have a set of state variables, and they evolve based on some differential equation of time. And there are there are some parameters, P1 to Pn. They can be in effect controlled, and they can be manipulated. and this condition is just a steady state condition. In the steady state condition, assume that the value is 0. Normalize it, shift it so that it's 0. So the setting for the modular response method is that we do not know the nature of the function f, but there is some prior information about this kind of about this nature is possible, that there are some parameters that do not affect certain variables. In other words, the partial derivative is 0 or not 0. So not the actual value, but whether it's just 0 or not. That's what is available. Not for every possible Fi and Pj, but for some of them. Okay, so the experimental protocol is this. Very simple. You perturb one parameter, say Pk. So when you perturb the state vectors, the concentration of proteins or activity levels of gene expressions they change. So you just let the system settle to a steady state, and you measure the state variables, and then you compute the sensitivity, which is the derivative, which you can approximate over a small range by the difference equation. Okay, so that's as straightforward as that. So the goal is that we want to obtain some information about the sign of these. So we cannot hope to measure the actual nature of these, the value or the rate. But what we want to know is that whether one state vector has a positive or negative effect on another state vector. Okay, so that kind of thing we want to know, if at all possible, from those experiments. So in a nutshell, if you do after some combinatorics and linear algebra, you can quantify the additional amount of information that you need. Okay, the amount of perturbations, that the number of perturbations that you approximately need, information theoretically to have the knowledge about this kind of the knowledge of how each state variable affects another state variable. But if you assume that you have sufficient information, or say close to sufficient information, then how do you determine actual minimum number of perturbation experiments that will give rise to the inferences that you are looking for? So now this becomes an algorithmic question, some kind of optimization question. So as I said, I am not going to discuss into too much of details for this. So this is a very high level overview. But after some effort, you can see that designing minimal set of experiments correspond to some kind of set multi-cover problem. So I'm not going to describe the set multi-cover problem or any such thing. So, but this is basically the overview of the idea. So this is the modular response method via differential equation. You have the linear algebraic formulation. Then you go through a combinatorial formulation where set multi-cover comes into picture. And then you have some kind of algorithm to select the perturbation experiment, which is going to be a randomized algorithm. And then you select the appropriate perturbation experiments, and then you do the experiments after that, and from there you can get the corresponding conclusions about the network that you're looking for. And there is also a feedback loop. If you don't like it, that you think that something in the network is wrong, then maybe some perturbation was done wrongly, or you need additional perturbation. Go back and forth and with the biologist until you are satisfied. So this is the overview of the system, and obviously I haven't discussed technical details of any of these. But there is an experimental analysis. So basically they are looking at the MAP kinase pathway, and these are the 3 proteins, and there are 2 receptors, and there are 2 NGF or EGF. These are the external stimuli. So you can control this external stimuli are those parameters P in the model. So you can actually control the stimuli, and thereby you can change the state vector to another steady state vector, starting from a steady state vector. So the MRI method was applied with respect to these 2 stimuli and these other proteins and other molecules as nodes to figure out the network. And it was done successfully. It's actually a very small network, I don't know, 5 nodes or 4 nodes or 6 nodes, something like that. So there is no need to use a computer. You can actually do all the perturbation, write down it by hand, and then you can figure it out. So this is like a proof of concept. But it shows that it's possible. So the other, yes.
A question because
Yes, sure.
you invited it. There was actually in 2013 a model published by Dennis Hickory with something like 50 components.
Yes.
And of course with experimental measurements and everything.
Yes.
Have you tried to apply your method in retrospect?
I kind of left this topic, so I am not working on it anymore. So. Yeah, I know there is some work, recent work out. In fact, there is another work by Barabasi. It's kind of controversial work, so let's not talk about it.
Huh? You've been videotaped.
Yes. I'll ask me later if you want to know. So the next thing that you want to talk about is decomposing a biological network into monotone systems. So this was a fun work to do. Again, we have this differential equation model. The state variables are evolving based on some differential equation. And for simplicity, we omit input and outputs, but it's easy to incorporate them. If you look at the paper, you will see that without input output, of course, no system makes any sense whatsoever. So monotone systems are a simpler behaved subsystem, like the dynamics is very nice behaved. So for example, pathological behavior, chaos ruled out. And in many sense, even though they may have large dimension, in many sense the dynamics behaves similar to one-dimensional system. There are no stable oscillatory behaviors, for example. So monotonicity is related to positive and negative feedback loops, and studying of feedback loops is of course a very well-known topic, starting with this work of Monod and Jacob. So the important thing to note is that interconnection of a monotone system may or may not be monotone. In some sense, positive feedback preserves it, but if you have negative feedback, destroys it. So the more advantage of studying monotone system is that you can characterize the behavior of an entire system based only a small amount of quantitative data, and there is no need to know the precise form of dynamics or parameters such as kinetic constants in the differential equation to infer many things about the dynamics of the network. So far so good. So the question that that we now need to address is that are biological systems monotone in dynamics, and if not, how close are they? So we again have the differential equation model, and from the differential equation model you can get something called the associated sign graph. So for every variable there is a node, and if the corresponding value of the derivative is positive or negative over all x, then you get an activation edge or an inhibition edge. Otherwise you don't get an edge because the behavior is not well defined. So and then which way is okay. And we also define a notion of sign consistency of the graph. So to note the sign consistency of the graph, think of the activation edges as plus, think of the inhibition edges as minus, and think of the total sign of a path as the product of these. So plus times minus times plus, whatever sign you get. So sign consistent graph means every path between two nodes have same parity. So this is not sign consistent. For example, if you take the path 1 to 4, it is negative. If you take the path 1, 2, 3, 4, it is plus, minus, and plus. sorry, 1, 2, 3, 4, plus, it is minus, minus, and plus. So if you multiply, you get plus. So you have two paths, one with positive and one with negative sign. So this is not a sign consistent graph. Whereas on the left side, the graph is sign consistent. So that's a very easy combinatorial definition. But what has this to do with monotonicity? So what one can show is that consistent graphs, if you take the differential equation model, build the corresponding associated sign graph, and if you look at the consistency of the graph, then you can show if the graph is consistent, the corresponding dynamics is monotone. So it's not both ways, but one way. So it's also easy to check that if a graph is consistent. You can just very simple modification of Lloyd-Warshall type shortest path. But what if the graph is not consistent, say almost consistent? So then the problem becomes, how many edges do we need to delete to make the graph consistent? So this is the combinatorial problem. So intuitively, it's like whatever edges you can take out, the remaining graph is consistent, and you can write the entire system as a consistent network followed by loops with negative feedback. So that's how you can express the entire system, which I haven't gone into details of that.
Isn't it true that for a graph to be consistent, all you need to take care of is, maybe not all, but at least you shouldn't have cycles with negative sign?
Yes. So it's very easy.
Like feedback arc or something?
No. We will see. The next slide, we'll see. It's not as difficult as feedback arc, easier. So actually what you get is an undirected leveling problem. So you have to level the vertices such that if you take the level of two vertex, add them modulo 2, you get exactly the level of the edge. When the levels are 0 and 1, and whenever you do addition, you have to do modulo 2. As simple as that. So it's actually simpler than the feedback arc model, because modulo 2 is easier to. So it's similar to max cut in some sense. So you can define the degree of monotonicity as the maximum number of consistent edges divided by the total number of edges, so that if the entire graph is consistent, it's 1. If you have to remove most of the edges, it is close to 0, and otherwise it's between 0 and 1. So there is a statistical mechanics interpretation in the Ising spin glass model. If you consider these positive and negative edges, then maximizing of the number of consistent edges is same as minimizing the free energy without any external magnetic field. So this is the algorithm for ULP. I'm not going to go in details. You have a semi-definite program, you then solve it, you have a random hyperplane. But the only thing that I want to emphasize, this can be easily implemented. It's not a utopian algorithm. It can be implemented even in MATLAB. And we have the MATLAB codes. You can download it. So
looking at me, I will tell you that you can probably also implement it with ILP and solve it to optimality.
Oh yeah, probably. But then you need a package for ILP. It depends on the size of the network, of course. If it is too, because we will show one large network. So it can be proved that you get almost 90%. So ILP or not, it really doesn't make much of a difference. And that's the performance guarantee due to semi-definite programming. So we took this network, which is the fruit fly Drosophila network, the signal transduction network model, which was published in Nature 2000, and this is the one cell level, and the neighboring cells are connected using those links, and it has 20 edges and 13 nodes. So our algorithm produced 17 consistent edges, and that's actually optimal. You can prove that for this particular network 17 is the number. So only 3 edges you can remove and you can make the system. So it's a monotone system with only 3 edges of negative feedback loop. You can write it. So this is just one cell. Of course, the real network is not one cell. So you can take many cell network, you can make it into an one-dimensional array by connection through those the two external outputs. And so our algorithm, so if you have a row of K cells, the algorithm produces 17K consistent edge, and you can show that that's actually exactly optimal. You cannot do better. So the other thing that we did was that we looked at the EEG-FR signaling network. So this is this network was published by Oda et al. It provided 330 molecules and 211 chemical reactions. So it's built from many sources, and you can get it in the SBML format. So what you do is notes the 330 molecules, and each reaction classifies the molecules, reactants, products, and enzymes. And so basically these are the edges. 0, 1, and undefined means that there is a conflict, and both of them happens. Fortunately, only 7 edges are undefined. So if you ignore them or somehow incorporate them in another way, you don't really lose much. So graph has 852 edge, only 211 reactions, so it's actually dense. It has dense subgraph. So this is how you ran the randomized algorithm and found 633 consistent edge out of the 852, which is not very large. So lots of edges have to be deleted to make it monotone. So it's not really very close to monotone, at least with respect to this algorithm. The question is that maybe this 633 is low because the algorithm was bad, or maybe it can be made even more better, right? So what we can say is that because of the performance of the algorithm, with very high probability, the actual number of consistent edges will be 728. So how close to 728 we can get? Well, so we finally are left out with 137. So if you subtract 137 for 852, that would be how much? 700 something. So those many consistent edges are there, and they're pretty close to optimal. So if you use another hybrid algorithm that you designed, not just the SDP, but SDP with some other algorithm, then you can actually get close to optimal. So with that, by the way, and for some reason this algorithm generated, where is it? Lots of quite a few papers in the computer science community, because apparently people could apply this particular combinatorial problem to some financial markets and some other places. And apparently there you get the same kind of combinatorial problem somehow, which I was not aware of. So the last part would be in signal transduction network. So we talk about 3 things: topological synthesis, minimization, and topological degeneracy and redundancy. So I'll just focus on the very last part and kind of skip over the first 2 because we don't have time. So the overall goal is that you have direct interaction, you have double causal interaction, additional information, then you need a fast method software, and you want to build a network that's minimal complexity and biologically very, very relevant. So there are various kinds of experimental evidence: biochemical, pharmacological, genetic evidence for differential stimulus. So some of these evidences correspond to a chain of chemical reaction. Some of these evidences correspond to a direct one-step chemical reaction. So, but sometimes you don't know whether it's a chain or just one step. Sometimes you know. Sometimes you know for certainty. So direct interaction is like this, and double causal or chain type interaction is that these 2 are affecting through some strange intermediate node that you don't know another node. So critical edge means that's an edge that you are with 100% sure that that is the right edge. There is no way it should be deleted from the network. So there is some notation that you get, and then you have 2 steps for network synthesis. One is pseudonode collapse, and one is binary transition. So this is polynomial time easy, and this is NP hard, and this needs a heuristic. And so pseudonode collapse, and so binary. So this is basically graphically what it shows, that If you have a edge and if you have another path with the same parity as the edge, then you just remove that edge. So something like that, intuitively. And you cannot remove a critical edge, never. So this is the overall goal. You synthesize direct interaction, optimize, synthesize double causal interaction, and then you optimize, interact with biologists. So biological validation, we took this plant signal transduction network. This is a network. This is a network for the stomatal closure of Arabidopsis thaliana, and manually curated, and they were, so the network was curated and constructed manually. So we wrote a software for this and constructed a network. So this is actually the manual curation, this is our curation, and our curation takes just 1 or 2 seconds, and basically it's a very similar kind of network. Whatever is the minor discrepancy is not unjustified. Oh yes, it's the previous one. It's where is it? Oh, it's it's not here. It's here. I forgot. I deleted that slice, but it's a slide. It's basically, it has like 100 nodes and some maybe below 200 edges. This particular network.
Right, but you need to insert these things automatically. You need expression data.
That is from the literature here.
How much? I mean, that's a critical.
That takes few years.
No, I know, but how much data do you need to confidently insert and make this network?
Just from those data. So these are a list of experimentally observable double causal relations. So you need this data and related data. So they have the interaction data. Some of the interaction are direct, one step. Some they don't know. It can be over a chain of biochemical reaction, and so forth and so forth. But the data is there, except that we had to correct a little bit some of the data based on our knowledge. But our work was minor. We were parasites in a previous. Who said you were parasites? Yeah. So the two are highly similar, and then if you want to look, you can download a software, just run, click. I don't maintain any server, and for Linux one, you can just ask for the source code, but there will be no graphics interface. And we also looked at another application of the software. We wanted to synthesize a cell survival, cell death in a large granular lymphocyte leukemia. So what we did was that we basically found there are 2 pathways that are important for the cell survival cell death network, and we want to concentrate on pathways and try to minimize the network such that we can focus on those pathways, and that can be done using a software. So in some sense it was simplifying the network, getting rid of unwanted nodes and edges while focusing on those specific fast ligand pathways which are important for this disease. So you can see further result in this direction. And then the last thing that I want to talk about is measuring degeneracy and redundancy. If you are an electronics or electrical engineer, then you know these concepts very well. Information theory also, in information theory also you know these concepts. And it's also known that biological networks do not have lowest possible degeneracy. For example, connectivities of neuron, they have a very high level of redundancy. So, for example, I use this code that although many similar examples exist in all fields of and levels of biology, a specific notion of degeneracy has to be firmly incorporated yet in the biological thinking, largely because of lack of a formal theoretical framework. And the other thing is also that there is a lack of effective algorithmic procedure. So you can define this based on the information theoretic measure, and you can go through mutual information of systems and define it. The problem is that the number of combinations that you need to do to evaluate this is, you can easily see, if n is 50, you have to go through 10 power 15 combinations. So what you do is that, so you can use the binary transitive reduction to Define, and then you can calculate redundancy and p-values for various networks, and you can say, have some conclusion based on the redundancy values for logical networks. Like one conclusion is that topological redundancy of metabolic network, the C. elegans metabolic network, is due to currency metabolites and so forth. So this is a slide that I put there, and I just out of my head is that whether you can apply spectral methods for studying biological systems, biological networks, or even phylogenetic networks. Like if you take the Laplacian of the graph and the eigenvectors, and what happens then. So some related publications which you don't have to read because it's impossible to read. And this is a book that we wrote, a shameless advertisement. But this is something that is very, very important, because without money we cannot do any research. And finally, these are the pictures of some collaborators, obviously, and whatever picture I could grab. As you can see easily that I look much better than all of these people. So with that, any questions that you have to ask?